Hello, everyone, and welcome to today's webinar, Post-Translational Modifications and Data-Independent Acquisitions, Challenges and Opportunities. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Cell Signaling Technology. To learn more about our sponsor, please visit them at www.cellsignal.com. Let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, just click on that support tab found at the right top corner of your presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located on the top right of your presentation window and follow the process to obtain those credits. I will now introduce today's speakers, Dr. Birgit Schilling, an assistant professor at Buck Institute for Research on Aging, Director of Mass Spectrometry Core at Buck Institute, an adjunct professor at USC, and Dr. Matt Stokes, a group leader at Proteomics Products and Services at Cell Signaling Technology. For complete biographies of our speakers, please visit the Biography tab at the top of your screen. Without further ado, please begin your presentation, Dr. Stokes and Dr. Schilling.
Okay, welcome everybody. Thank you for joining us today. This is Matt Stokes. I'm going to give a brief introduction to PTMScan proteomics and then turn it over to Birgit to talk about her work. So when we talk about PTMScan, it's really just the combination of an antibody enrichment with liquid chromatography, tandem mass spectrometry to both identify and quantify post-translationally modified peptides. And really the key to success in PTMScan are the antibodies that we're using. So typically when we develop an antibody, we use a single peptide or a piece of a protein or even a full-length protein as the antigen. Here we're showing a phosphopeptide. The antibodies that we use for PTMScan, however, are a little bit different. So instead of a single peptide sequence, we actually use degenerate peptide libraries. So here again we see a phosphopeptide. It's a phosphoserine residue with an arginine at -3. And every peptide in the library will have those two residues fixed. Then we vary the amino acids around it. So we can do this to make phosphomotif antibodies. We can also use it to make antibodies against other PTMs, such as acetylation of lysine. So we use these specialized antibodies in the method, and one of the great things about PTMScan is it's very flexible as far as input material. So you can use cell lines, tissues, we've done plant material, microbial samples, whole organisms like Drosophila, sorted cells or subcellular fractions, even serum or plasma. And one of the first things we do is digest to peptides. So here we're doing peptide level enrichments, not enrichment of full-length proteins or protein complexes. So we then enrich with one of those specialized antibodies, take that enriched material and run it in mass spec to get identification of our peptides, and then we can do quantification, looking at what's up and what's down across the various samples. And we're able to assay a lot of different PTMs using this method, things like phosphorylation, acetylation, methylation, other lysine acyl modifications. And I wanted to tell a brief story today about two of these in particular, ubiquitination and simulation. So this is just a picture of a ubiquitinated protein. The target protein is in the middle in blue, and the ubiquitin is on the left in green. And you can see you have attachment of the ubiquitin to an epsilon amine of that lysine residue in the middle of that target protein. When we take and digest this with trypsin, what we end up with is, of course, a peptide from that target protein. But any place where we had a ubiquitination event, any of those lysines, we actually end up with a diglycine remnant. And so we made one of these PTM antibodies against that diglycine hanging off of a lysine. So you can take your samples, trypsin digest, enrich with that antibody, and now look at thousands of sites of ubiquitination in samples. And we've been doing this for ubiquitination for many years, but we were never able to do simulation. And the reason is In ubiquitin, you have an arginine next to that diglycine, so it's cleaved by trypsin. But sumo, be it 1, 2, 3, or 4, actually doesn't have that arginine. It has threonine residues, so you can't cleave it with trypsin. So ideally what we wanted was an enzyme that would cleave after threonine residues, but not at arginines. And that enzyme actually exists. So this is some data from a paper a few years ago in Molecular Cellular Proteomics, where they were characterizing this enzyme alpha-lytic protease, or WALP. And it cuts at a variety of residues, and importantly, it cuts at threonine residues, just after threonines. The other important thing here is it actually doesn't like to cut at arginine residues. So now we have two different enzymes, and we can take the same sample, split it in half, cut half with trypsin to look at ubiquitination with that diglycine remnant antibody, take the other half and digest with the alpha-lytic protease, and now look at sumylation. So a little bit of sample data on this. We took some HeLa cells and left them untreated or treated with desumolylases. And so these top two Western blots here with SUMO1 or SUMO2-3 antibodies. And what you can see is when you treat with those desumolylases, the signal goes down in those blots. So we take these same samples, cut with the alpha-lytic protease, and run the mass spec. And what we see is basically all of the peptides are higher in the untreated sample than in the desumolase treated. Right? Everything is to the right of 0 in this histogram. We can take that same sample and now blot for ubiquitin, just a total ubiquitin blot. Here, of course, we don't see any change minus plus those desumolases. And likewise, when we now trypsin digest that material and run the mass spec, there's essentially no change in ubiquitinated peptide abundance between minus plus treated. So a really nice example of the kind of specificity of the method and the fact that you're looking specifically at sumylation when you do this alpha-lytic protease digest. So we have this available as a service, and some exciting new news from us. We've actually worked out production on a large scale of this enzyme, and so we are going to be able to start selling it as a kit very soon. So with that, I again wanted to thank everybody for taking the time today, and now I'm going to pass the mic over to Birgit to talk about her work.
Well, thank you, Matt. So this was quite a nice introduction, in fact, then for my talk, because I will show you a little bit data how we are using these tools that Matt just described and really apply them to relevant biological projects that we are performing here at the Buck Institute for Research on Aging. You'll see a picture here. So on the second picture, you can see that we're our mass spec core and our instrumentation. We do have several high-resolution instrumentation for us available. And as you can imagine, when you have a core, you may have a lot of very different projects. Here is like a short overview of some of the projects. So some, look at breast cancer, others look at more biology, concepts of aging, other projects look at bacteria or biomarkers or Alzheimer's or Huntington's. So, the variety of projects can be very broad. But what is really interesting is that a lot of projects actually have, post-translational modifications involved. I mean, some people may only want to look at protein expression, but again, a lot of signaling and very dynamic aspects in the cell can happen via post-translational modifications. So we have in particularly a lot of projects that are dealing with, acylation, and that may be acetylation or succinylation or myelination. And what is interesting is that there are acetyltransferases that add the acyl group onto a protein, and then there are also deacetylases that can remove it again enzymatically. And, this always reminds me, this is maybe similar to what you see in phosphorylation, where you have kinases and phosphatases. So many projects that we have, in fact, rely on enrichment strategies for PTMs, as the PTM levels often are relatively low when you just look at the lysate itself. So we have been working with CST antibodies for affinity enrichment for over a decade already, and we're even involved in some early development for acylation together with Dr. Worden and CST. What is interesting is that, Matt mentioned there are these now new PTMScan antibodies, particularly against acyl groups here for acetyl, malonyl, and succinyl, and we have actually used them. And one thing that's why we like these in particular is that they have a really high enrichment efficiency. We often see between 60 and 80% of then just pure acyl peptides versus other non-modified peptides. So that has worked out quite well for us. And I would, and actually Matt had shown you there's a suite of phosphorylation antibodies also available. Actually here, a paper from Matt. But I wanted to show you some of our own work that we have done over the years, and particularly looking at acetylation here. So I mentioned to you there are ways how to add on an acyl group and also to take it off. So deacetylases actually they remove the acetylation group, and when you knock out the deacetylase, you can enrich, or you can kind of increase the level of acetylation. And then maybe when you compare that against, say if you have a sirtuin 3, that's the deacetylase knockout, and if you compare that against the wild type, you can see which particular sites the sirtuin actually regulates versus which sites are already there in the wild type and are not regulated by the sirtuin. So here you can see one of our typical workflows. We digest, we take cells, isolate mitochondria, we digest the proteins, and then do the acetyl enrichment with the PTMScan antibodies here. And then do LCMS and look at, either do MS1 filtering, or more recently we've switched over to doing data-independent acquisition, DIA or SWAT, which is actually quite comprehensive. And, so the combination of these really very efficient enriching antibodies with, modern mass spec scan types, I think that has allowed us to get deep insights into the biology of these systems here, the role of sirtuins in this case, for example. One thing that is actually very interesting we find is that, there's a very dynamic behavior of acetylation or PTMs in general. On the left and as well on the right also, you can see that if you take, say, one protein on the left, isocitrate dehydrogenase, and then monitor different acetylation sites, here indicated by different color curves, and on the x-axis you'll see, say, a time frame, like a time course, you can see the very different dynamic response of PTMs even within one protein. So you have to really kind of treat each acetylation site as its own, because you can see some, they start out slowly and over, at 2 hours have increased a little bit, and when you have different conditions, for example, of glucose supplements here versus control where you don't have that supplement. And, so you can see some increase slowly and others kind of skyrocket and have very high fold changes, 10-fold changes. So it's really important looking at the individual acetylation sites, which is a very different mass spec question versus if you look at protein expression changes. But I wanted to show you a couple of different examples, and Matt referred to this earlier. These antibodies, they work on mouse, they work in bacteria, and actually here I wanted to show you one slide that we work a lot on protein acetylation in bacterial background. And it's actually interesting because in bacteria, you have different mechanisms. There's acetyl-CoA, and there's also something called acetyl phosphate, which actually doesn't exist in higher organisms in mammals. But what is interesting is that, you can achieve acetylation through our traditional way of having an acetyl transferase. They call it here a PAT or CAT, lysine acetyl transferase, where you have the acetyl transferase and the deacetylase, and you use acetyl-CoA to transfer the acetyl group. But in bacteria, there's also the acetyl phosphate, which can do chemical acetylation. So that does not need an enzyme to do this acetylation. And so you have multiple mechanisms happening in parallel and at the same time, so great grounds for regulation of the bacteria. One project was we wanted to find and identify new acetyl transferases. And you know, I mean, you can look at the genome of, say, an E. coli organism and predict which proteins may be acetyltransferases. But then it had become, you can then overexpress them and see if they regulate acetylation. But there was a lot of background in bacteria. So we actually tried to, you know, knock out a lot of these kind of mechanisms. If I go back to the last slide, that generate acetyl phosphate, we can knock out the copy, for example, and other kind of aspects that are part of the machinery. So we're really looking only at the pure acetyltransferase candidates. So my collaborator would make these knockout strains, we call them the gutted strains, that have very low level wild-type acetylation levels, you know, particularly not having the acetyl phosphate mechanism there. And then you can try and overexpress these potential cuts. So they made 22 strains, and we would in fact you know, monitor all these strains first with antibodies, Western antibodies. And these are actually also cell signaling kind of antibodies that are made more for Western blotting. So you can see that if it overexpresses like a potential cut, and in the anti-acetyl Western blot you don't see a signal, that's not really an interesting target for doing mass spectrometry on it afterwards. What was nice, this was a known acetyltransferase, and you can see how that has like a really nice, you know, acetylation pattern over, you know, say your vector, you know, your control setting here. And then you can see there were some transferase candidates that were more promising than others. So we chose those and now submitted those for our mass spec workflows. And, you know, particularly the ones I'm highlighting here, these we selected, and we had like a set of, out of the original 22, we now selected 5 acetyltransferase candidates based on the Western blot analysis, and we had the what is called VC, the vector control, that didn't have an overexpression of an acetyltransferase. We would digest, and we actually would use different enzymes here, either trypsin or gluC, but in separate experiments, and then use the CST PTMScan antibodies to do the enrichment, and then subject to data-independent acquisitions and quantification. So with the data-independent acquisition or SWAS, we can do identification of the acetylation sites in parallel and at the same time as the quantification. So that was kind of a nice throughput we could do on the mass spec end, because we had quite a few, you know, cut targets as well as biological replicates, so the number of samples would increase quickly. On the next, and again, here you can see, you know, the SWAS type data here. We would, you know, do the quantification based on fragment ions. These here are peak areas plots. And you can see, for example, for this particular peptide shown here on a protein called signal recognition particle protein, you know, some of these cut candidates would show a really high increase in acetylation levels and others not at all. So there was some exciting specificity that we could see here. But overall, you can see that in these systems, we were able to see many acetylation sites that we could identify. In this experiment, in fact, over 2,500 unique acetylation sites. And actually, I'll show you later some slides. If we also digested with other enzymes, other proteases, such as gluC, we could actually expand the portfolio of PTM sites by a multitude, here even going up to 3,000 acetylation sites. And the reason for that is that one just cleaves at different, you know, chemistry. Here at the lysine, I mean, either you do trypsin, lysine, arginine, or gluC, aspartic acid, and glutamic acid. So you cover other areas of the protein. What I show in the Venn diagram is actually that these different cut candidates have very different site specificities, and the overlap would be, you know, there, but there are many unique things that are only targeted by some of these specific acetyltransferases. This also is demonstrated in the next slide. where, you can see, where some of the cuts would have specific targets for some of the acetylation sites in the given proteins here. Other acetylation sites would be more global and could be regulated by several of these acyl transferases. So this was actually quite interesting, and again, I wanted to emphasize that this project was really only possible by having a good enrichment and an efficient enrichment and a reproducible enrichment strategy, because we were also working in a label-free environment, so everything has to be really reproducible. It was interesting to see biologically that many of the central metabolism was affected here, and the little dots show us either in red or in blue in the tables indicated which of the cuts had specificity for these sites. So this was biologically rather interesting for our collaborator. And you can imagine many follow-up experiments on a project like this. You can do in vitro incubations. You can look at enzyme kinetics. We have purified these cuts now also on a preparative scale. We connected with Sean McSweeney at the Brookhaven National Lab for crystallization of some of these cuts, as well as substrate proteins that are acetylated, so crystallizing acylated proteins. But really, we needed the mass spec data to even know which ones are targets, which ones are, regulated. So, I hope I could show you in this E. coli project how we used affinity enrichment together with the DIA, data independent acquisition, as a very efficient way to deeply mine the acylome in these bacteria. As a second part, I'd love to show you some data that we did in mice. So these antibodies also work really well in mice. So we have acetyl-CoA, and you can now imagine, use this acetyl-CoA to either chemically acetylate or also use acetyl transferases in these mammalian systems. The systems we were looking at were actually kind of interesting. Well, it looks maybe now, I don't know, the image may not, what's happening here? The image doesn't look as good here. I'm not sure why that is the case. But this is supposed to show you like a liver histology. And, we were looking at different conditions here in feeding. So maybe I can just rather show you on the next slide. I'm not totally sure why can you see my slides? I'm not sure. I'm having a problem seeing the slides here. But in fact, it doesn't matter too much. Basically we were having different acetylation or like feeding regimens. Let's go to the next slide. Yeah, and so basically we were feeding these mice acetylation, succinylation, and then also we're looking at the acetylome, the succinylome, and also at the protein expression changes. So we had 6 dietary regimens, fructose, glucose, high fat, and then combinations thereof. And then 5 replicates for each of the conditions. So we had overall 60 samples, and so that was quite a large experiment here. So we would process everything with swaths here, and that was also quite nice because we would be comprehensive in the mass spec nature. So, okay, we switch to the next slide. Here on the next slide, you can see our workflow a little bit. And so we would again, digest the samples, get a mitochondrial extract, and then digest the mitochondria. And then, in fact, use the antibodies to enrich here. And we would start with acetyl, then do succinyl. So basically, we would use 2 separate samples for that. And, in fact, we would use, the different, some different workflows here we call DIA umpire to then analyze our samples. So DIA Umpire is actually a module that was developed by Tzouros, and he, you can basically search DIA data by themselves. So you can actually make pseudo spectra where you match up fragment ions to, your precursor ions, and then kind of get a pseudo MS spectrum, and then be able to search that in with your typical data search engines. So okay, all right. Oops. Okay. So on the next slide, we have developed here a workflow which we actually call PEAKed, and that is an automatic processing of these data-independent acquisitions to do database searches. So you can use the data-independent acquisitions to do your searches, and then at the same time do quantification. And so let me see. On the next slide, see here, this shows you, like a little bit of display how this may look like. And in the end, when we do this, we would basically get like an entire, kind of portfolio of looking at proteome changes as well as acetylome changes and succinylome changes. One thing that I also had mentioned in the abstract, what is actually really interesting is when you do have post-translational modifications, you could actually have a post-translational modification maybe on one site or another site in the same peptide. As I'm indicating here you see an example where you have actually a phosphorylation site either on the serine 293 or on the serine 300. And when you do data-independent acquisition, you'll actually get both spectra, and you will get, say you can look for distinguishing ions here, in this case the Y6 ion, and you can determine based on your mass spec data whether the localization was on one or the other serine residue. So this is actually quite interesting and has become something important in our lab, and we have actually developed some algorithms to differentiate these isomers and to quantify them independently by choosing these ions that are specific for one or the other isoform. So we actually generated some, kind of synthetic peptides here, these two peptides that I'm showing here, and mixed them in at different ratios as I'm showing you on the next slide, and then basically tried and see if we can get a linear signal when we measure this with mass spectrometry. And in fact, in this case, we actually were able to show this and demonstrated we spiked in different levels of these synthetic peptides from 1 to 16 to 16 to 1, and we achieved quite a reasonable linear curve here. So, that was kind of important, and, a lot of effort now goes into really this differentiating of different isomers in real-life samples. So we're doing a little bit technology development on these isomer identifications. At the same time, we are also combining this with a new workflow that we have recently published on, and it's kind of funny, we call this like the multiple PTM affinity enrichment, or, also in the lab we call it the one-pot enrichment. And what that means, instead of using just like, say, one of these PTMScan antibodies, say first an acetyl antibody and then a succinyl antibody, either from different tissue starting material or from serial, enrichment, we would just mix all the antibodies in one, tube, and in one pot, in fact, and then see how, do the enrichment from one piece of tissue and enrich at the same time for acetylation and succinylation. One advantage in this is that if we are using, say, samples like mitochondria from mice, or maybe like kind of the cortex of a Huntington's mouse or the striatum of a Huntington mice, So sometimes you're actually rather sample limited in the starting material. So the one-pot enrichment really allows you to start with, one set of starting material and then do all your enrichments just from that. So that has been in the lab a big advantage, but of course it also cuts instrument time in half and sample preparation in half. In fact, actually, I'm showing this to you here. I mean, if you do a serial enrichment where you take the workflow of, what comes, the leftover of the acetylation enrichment, if you take that, it doubles the time. Or if you even start from two separate sample sets, two separate tissue sets, it also increases the work, the work that you have to put in, or the research associate or postdoc in the lab. So, but we had to assess, well, is the efficiency the same? Is it, giving us the same amount of sites? Is it giving us the same quality of data when we quantify? And so we actually assessed this here a little bit. So here, in these correlation slides, you could see, well, let's see if we do a one-pot enrichment, is that the same quality versus if we do just like a single individual enrichment? And we were also testing these sequential enrichments where you take the workflow. So we did a lot of different tests and assessed really not only the number of acetylation or succinylation sites that we would obtain, but also the quantitative efficiency in those. And as you can see here, we had actually very good correlations, and for us determined that the quality of data was just as good as if we did individual experiments. What was actually quite interesting and, was that we, actually, let me go back to this slide, that the overlap between the different, the acetylation and succinylation sites was sometimes quite unique and different. So, here you can see, for example, if, we certainly for the lysine sites, we had 943 lysine sites that we found both acetylated as well as succinylated. But there were also like a large number of sites that maybe we only observed as succinylated or only observed as acetylated. So that was actually quite interesting. We also assessed whether, here you can see this actually shows you fold changes in specific proteins here, PDH or BCKDH or ATP synthase. On the right, you can see, kind of the, log Q values here, but also the Q values and the log fold changes. What was really interesting in this particular experiment from the mouse mitochondria, we actually would see many sites on complex 5 of the electron transport chain complexes. In red were subunits that we saw either succinylated or acetylated. And really, I think one advantage of this PTM crosstalk and one-pot enrichment was that we could see these different modifications together. And not maybe, you could imagine that maybe you lose some succinylation sites if you do the individual acetylation enrichment, and you may miss those. In the one-pot approach, you would see them together. In fact, in the data, it was actually quite interesting. We would get all these spectra here, for example, where we would have mixed species. Maybe there was one peptide that we saw both acetylated as well as succinylated. But we saw the same peptide also where both of these lysine residues were succinylated. So we saw all kinds of really interesting, different portfolios of acylation sites on one and the same protein. So we can maybe really try and look at crosstalk of PTMs here. And in fact, here I'm showing you one example where we actually were able to differentiate which sites of these lysines here were modified. There were two lysines right next to each other, and we could actually find differentiating ions that would be able to see was it succinylated on one or the other lysine residues. And in fact, we also saw these mixed species here, and then also where was the acetylation site and where was the succinylation site. So using DIA and SWOS, as you are monitoring all fragment ions at the same time, we're able to extract these differentiating ions and then do quantification based on those specific ions. And in fact, I'm showing you this here. Here on the left, you can see, like a total ion chromatogram extracted here for the different fragment ions here. And the Y7 is an ion that differentiates here that on the left, you have the succinylation site on that lysine. And if you look at the panel B, you actually have a different peptide species that elutes a little bit later, and you get different M/Z values for these Y7 ions in this particular case. So we would have even been able to differentiate these isoforms if they had co-eluted. So this is really a power of combining, really very well done affinity enrichment tools and workflows here with modern scan types and really getting the most information out of a PTM space as possible. And I mentioned earlier to you, you could actually increase your coverage in depth even further by using different proteases. We showed in this particular project that when you do trypsin or digestion or glu-C digestion, which more cleaves at these acidic residues, you get a whole different set of PTM sites shown here, either in blue or red. And this is the site level, and the same is also true when you look at the protein level here. So for example, let me go back to the last one here. So yeah, see, this is the protein level shown here. So even on the protein level, you cover new proteins that you never saw just using trypsin digestion. So when it is important enough, we actually do use this approach of different proteases. So we feel we get a really deep insight into PTM space. And this was actually true also for this other project that I showed you earlier in the E. coli data, where we were looking for cuts. We used different enzymes here, trypsin and glu-C, and were able to see a whole different set of PTMs that were not even detected before. And the reason for that was purely that it didn't generate a well-ionizing peptide. So there are a lot of chemical and biochemical tools in order to look at PTMs. This is my last slide, kind of summarizing the data. So we have really become interested to look at PTM isoforms and crosstalk. We use this one-pot enrichment in order to cover more than just one modification at the time. And we often now use DIA SWOS workflows in combination with quantification in Skyline, an open source software, to get the most out of our quantification data, and also to be able to easily visually access the data that we achieve and acquire from the instruments. So I think what I have shown you today is a big interest that we're having in PTM, dynamic aspects of PTM research, and getting as much depth as you can. And the PTMScan antibodies were a great tool for us in all these projects and have allowed us to really find interesting biological events. I'd like to close with showing you my group here at the Buck Institute in California. And I'd like to thank members of my group and of course also CST, my other collaborators, and kind of our grant support. And I'd like to show you a recent picture from last year from the Buck Institute here. And I'd love to open this for questions, and I'll be happy to answer anything you'd like to know about our workflows or other mass spec, kind of experiments that we do here. Thank you.
And thank you, Dr. Stokes and Dr. Schilling, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's take a look at our questions. Our first question is a two-part question. Dr. Schilling and Dr. Stokes, are you considering including more post-translational modifications in your one-pot approach, such as ubiquitination or methylation? That's the first part of the question. And the second part is there a limit to the number of modifications it would be appropriate to combine?
Okay, so maybe I start with answering this. So yes, we definitely have a high interest in expanding the portfolio of one-pot enrichment antibodies, or adding other PTMs to it. In fact, the ones that was asked about specifically ubiquitination and methylation, those would actually fit very well for adding them into the one-pot enrichment. The reason being because the only thing one has to consider is that the workflow and the kind of solvents and buffer, that works for all the antibodies that would enrich the specific PTMs. As particularly here for ubiquitination and methylation, as Matt mentioned earlier, CST antibodies are also available, and they actually use a very similar set of conditions to perform the enrichments. So that should actually work very well, and we have actually started with some pilot experiments doing this. So I think this is actually very interesting, adding more to it. The second part of the question was, is there a limit to the number? Well, I mean, in theory, it shouldn't really have a limit. The one thing that one might consider is just the case that maybe at some point the mixtures do not get too complex, because one reason why we do these enrichments is that, we can focus most of our mass spec time on the enriched PTMs. But on the other hand, that is usually not such a problem. When we look at PTM-enriched fractions, they're usually really not that complex, and with the modern mass spectrometers, they are so fast scanning and have such good sensitivity that I see this to be actually a really interesting trend for the future.
Thank you very much. And are you generally able to confidently determine the site of modification?
Thank you. Yes, so I mean, using these DIA workflows, we found that has really improved our ability to identify the site of modification, because when people do data-dependent acquisition, what often may happen is that you trigger the MS-MS spectrum on one isoform, and then there's dynamic exclusion for that particular M/Z precursor ion. If one does use data-independent acquisition, everything will be monitored all the time as part of, what the concept of data-independent acquisition is. So that has helped greatly. I will have to say, for the acylation, this has worked actually extremely well. It is sometimes a little bit more challenging for the phosphorylation. As you all know, sometimes there are stretches where there's multiple serine or threonine, residues right next to each other. Then it does become more challenging. But in many cases, this actually is possible.
Thank you. And another question coming from our viewers: would the DIA one-pot method be appropriate for phosphorylation analysis?
That is also a very good question. And, so for example, what was, I thought, as a user, very impressive for me was also to see the portfolio of phosphorylation enrichment antibodies that Matt showed earlier in his presentation, focusing on phosphorylation enrichments for different pathways and, the serine and threonine and tyrosine antibodies. So yeah, I do think that one could also combine these particular tools. We haven't done this for the phosphorylation yet, although I have to say in the past, other groups have done this kind of work, combining phospho-enrichment methods. So I actually do think that this is just equally possible, and it might even be interesting to try and see if one can combine different classes of PTMs even. So I do think there's a lot of, space to explore these things and really look at these PTM crosstalk and, I think dynamics in particular.
Thank you. Our next question coming in, would you expect similar results using a data-dependent scan type?
So, I mean, I think for, you know, getting the very high numbers of PTMs that I showed in some of my presentation earlier, for that definitely the DDA will also be, I think, a very good way in order to work with, I mean, having a workflow of doing the, you know, enrichment and then the DDA identification, or maybe then TMT quantification for, you know, but what I was saying for if one really wants to look more or specialize also on the site localization, I do think that maybe the DIA has some advantages over it because of the comprehensiveness and maybe the ability not to lose very minor isomers that may be present. So, you know, I mean, we in our lab, we have shifted over a little bit more tending to the DIA approaches. You know, that's just the preference, I guess, in our lab, right.
Thank you, and I want to thank our live audience today for their questions. We have time for a couple of more. Would this approach work for serum samples or other sample types?
Yeah, I think, you know, we have actually a lot of different samples in our institute. We sometimes also get like, you know, like C. elegans samples or Drosophila. We have like a broad spectrum. I have in the past worked with a serum, or actually rather plasma, and we did do a lot of glycosylation analysis. Yeah, I do think, you know, that this particular workflow and the PTM, you know, one-pot workflow and these antibodies, they should just work fine in plasma. In fact, this is something that we are also, you know, intending to do with some kind of longitudinal samples that we will get from plasma soon. So yeah, that should work just as fine.
Yeah, and I'll just interject here as well. I think, you know, one of the cool things about doing the PTM scan in a serum or plasma sample is what we've found and published on is that it kind of obviates the need for depletion kits or depletion columns from that serum. So you can directly take that serum. You know, a problem with serum are the abundant proteins, things like albumin, the other 20 abundant proteins. You can take that sample now and just do, say, an acetylysine enrichment or a methylarginine enrichment. Now you don't have to deplete before, and you're getting a real good kind of look at even lower abundance proteins in that serum by doing the enrichment.
Thank you, Dr. Stokes and Dr. Schilling. We have time for one more question from our viewer. If you compare AB-enriched samples versus label-free acquisitions of non-enriched samples, do you see modified peptides that escape the enrichment due to PTM context not being optimal for AB detection? And if so, how do you deal with such biases?
Yeah, that's a very interesting question. I do think that it's probably true that one will miss some, you know, PTM sites. I think any given workflow will have a certain strength and maybe, you know, something that one may miss. So, I mean, and also you cannot exclude, but I mean, I think the dynamic range enrichment that one does get through the affinity enrichment, I mean, that is a specific strength that I do believe, you know, really contributes to identifying that many sites. We have actually worked with somebody before, Sam Payne is his name, on some of these E. coli projects and bacterial projects. They have actually done analysis just out of the regular lysate without enrichment. But, you know, I mean, for us also, what is really important is not just identifying something, but it's mostly that we want to quantify them. And when we do these enrichments, the space and the matrix is so much cleaner because you really only say you have 60 to 80% of these PTM-enriched peptides now available. So the quantification that we do afterwards in Skyline from the DIA acquisitions is so clean and has so little interferences. That that is really important to us because the quantification of different conditions, that really brings in biological interest. However, we have actually talked with some other researchers where we have now built some of our libraries out of the enrichment strategies, and now we have given our libraries to some of our colleagues who then use these given libraries in DIA mode and look for these peptides that we now know are there out of the entire lysate, without enrichment. So I think combining these different workflows and intelligent or smart design of the experimental, aspects, I mean, that's kind of, you can get a lot of information out of that. And I think there's a certain strength in having these different workflows and just knowing what is my focus and what do I want and what's most important to me.
Yeah, so I think Birgit is absolutely right, and I would just add to that whenever we're preparing one of these PTM or motif antibodies, our goal is always kind of the broadest coverage possible, or to generate something that really is context independent and is really only dependent upon presence of that PTM. So all of these products are monoclonal products, but what we'll actually do is take clones that we get, test them via ELISA, or actually test them in PTMScan, and look at the peptides that we get out of each of those individual clones, and in some cases combine multiple monoclonal clones into one product. A great example is the acetylysine. So that's actually a mix of 7 different monoclonal clones, and we did that to really maximize coverage and get one reagent, one tool that should, in principle, be able to pull down any lysine acetylated peptide.
Dr. Stokes, Dr. Schilling, thank you for your presentation today. Would you like to provide our viewers with some closing remarks?
Oh, yeah, sure. So, I do really think that it is very interesting having these developments, and particularly taking advantage of the specificity of these antibodies and really using them and applying them to a biological project and having maybe a hypothesis, maybe, okay, I do have some evidence that the PTM space here changes, and, having this combination of a smart workflow and sample preparation that's really highly reproducible with an interesting biological question, and then, as I mentioned, the appropriate mass spec scan type, and I think that really can help the biology collaborator to get the most out of their precious biological material and gain biological insights, which is really the ultimate goal of the experiment.
And from my end, I would just again thank Dr. Schilling for sharing her work today. CST is a company of scientists for scientists, and it's really always exciting for us to be able to see our products be put into action and doing the kind of cutting edge research that she is doing.
Thank you.
Well, thank you both, Dr. Stokes, Dr. Schilling, for your time today, and again for your important research. We'd also like to thank LabRoots and our sponsors, Cell Signal Technology, for underwriting today's educational webcast. Before we go, I want to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via contact information that you provided at your time of registration. So this webcast can be viewed on demand through April 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share this email with your colleagues who may have missed today's live event. That's all for now. Until next time, bye-bye.